Thank you all so much for attending our latest thought leadership event. This is the second in a new series of talks here at CRDF Global meant to highlight important ongoing global concerns and the discussions towards their potential solutions. We want to welcome anyone in our online audience to go ahead and throughout the entire talk, submit your questions to the Q&A section in the webinar settings at the bottom of your Zoom. And we will be happily passing those on during our Q&A session that will be immediately following our talk from our amazing speaker today. The issue that we want to highlight today is genomic data security. As our lives move significantly more online, so does research and scientific data. These data are vulnerable to attack from state, non-state, and lone wolf actors who seek to utilize these data for nefarious purposes. The greatest of these threats is the weaponization of genomic data for the creation of biological weaponry. Genomic and bioinformatic data, broadly from viruses, bacteria, and other pathogenic organisms with the potential for human loss of life, have been a growing target for malicious actors. For example, researchers have long hypothesized that it is possible to synthetically recreate viruses such as smallpox based on available data on the eradicated virus. We also know that institutions like universities have been the subject of ongoing hacking events. It is not a far leap that a future is possible where a malicious actor gains access to bioinformatic data and uses that to create a weaponized pathogen. For this reason and much more, it is integral that we strengthen the bioinformatic infrastructure against these attacks. CRDF Global's vision, mission, and values have been driven by the shared understanding that threats and opportunities often go hand in hand. CRDF Global is currently working on several projects, including one you'll hear a little bit more about today, on managing the risks and rewards of genomic data and to ensure scientific advancement for societal good. Our work mitigating risks associated with genomic data has led us to collaborate with GeneInfoSec. GeneInfoSec's mission is to protect sensitive genomic information by securing bioinformatic data through a process they will call and name as molecular cryptography. This obscures genetic data so it cannot be traced. Today's speaker, Dr. Sterling Sawaya, is the founder and CEO of GeneInfoSec and a leader in the field of biosecurity. He received his PhD in genetics from the University of Otago in 2013 and has since utilized his background as a scientist to tackle some of the largest issues in biosecurity. He has appeared before the Bipartisan Commission on Biodefense, is on the Consulting Advisory Board, American Co., and a 2020-2021 Fellow of the Emerging Leaders in Biosecurity at Johns Hopkins University Center for Health Security. Please join me in welcoming Dr. Sawaya.
Just briefly to outline the talk today, I'm going to cover some basics in genetics and some background so that everybody can be on board no matter what your education level. I'll then be discussing the current situation around genetic data, where it's going, who's collecting it, how it's being collected, and discuss some of our research on genetic information security itself related to lab cybersecurity. And then, of course, we're excited to also talk about the technology we're building at GeneInfoSec. Next, please. So some basic background in genetics. It's a very young field. So it wasn't until 1865 that Gregor Mendel discovered heritability in the form that we understand today. And in fact, he was not recognized in his time. And he studied these, the humble pea plant that you see there. He studied the color of it, the height of the plant, and found this interesting form of inheritance that wasn't exactly a blending between two parents, but rather was, represented specific alleles that combined in ways that could be dominant or recessive. Then decades later, DNA itself was discovered as the actual molecules that allowed for the transmission of information through generations. And then another 25 years later, we actually understood how that molecule was structured, that it was in the form of a double helix. So that's really not that long ago. You can realize that not long after we found out about the double helix, we had people in space and men on the moon. Then 1977 was when we first were able to sequence DNA and determine those A, T, C's, and G's. But that sequencing was very expensive. So we didn't sequence entire genomes and we didn't really understand how the DNA was working, but at least we could get some insight into it. We did sequence smaller genomes along the way, but one of our biggest surprises came with the Human Genome Project in 2001, where we spent billions of dollars to sequence a full human genome. And what we found was that genetics didn't appear to work like we thought it did. We had sequenced a worm previously and found that a worm had tens of thousands of genes. We thought, well, humans are more complex. We must have hundreds of thousands of genes. But in fact, we only had about the same number as worms. So still today, 20 years after we've fully sequenced the first human genome, we're still really understanding how that genome works. And again, comparing that to something like physics, where we've been collecting data for decades, or chemistry, where we've had hundreds of years to play around with these things. Right now, we've really only been in genomics for a couple decades. And now it's starting to accelerate. So a few years ago, a full human genome cost $1,000 to sequence. Next slide. That's incredibly less expensive than the billions of dollars it costs to sequence the first human genome. So the actual cost of sequencing full human genome is dropping faster than exponential. So the y-axis there is a log, so that line is an exponential drop, and the fact that it's dropping faster than that means it's faster than exponential. So we're expecting potentially as soon as next year for that next drop to occur, where we'll be able to sequence a full human genome for around $100. And from there, again, it's expected, okay, will it take 5 years? Will it take 10? But then sooner than later, it'll be $10 for a full human genome. That's almost nothing. So we'll be getting all this free data, and it's going to be incredibly useful. And as a geneticist, I love science, and I love the fact that we're collecting this data. But we should be aware of some potential downfalls. Some basics around genomics. So we get one copy from our mother and one copy from our father as mammals, and there can be differences between them. So one difference that's very commonly examined is called a single nucleotide polymorphism, or SNP. I'm going to be referring to it a fair bit in this talk. And it's essentially just a single letter change. So you can see the G to the T or the G to the C. And that type of information has been, up until now, sort of the cheapest way to collect genetic information. We can use technology that collects SNPs all the way across your genome, so we can examine millions of them, and it's relatively inexpensive. But it doesn't capture all of the variation. This is somewhat important because there's other variation in the genome. And so you can see there that there's this other information there, this other variation that is incredibly important. That's now captured through DNA sequencing. So a lot of the information that we've been getting so far has been snapshots, SNPs of the genome, not the full picture. So, we've got tens of millions of DNA tests done. This is a lot of this is in academia, but most of this is in industry, collecting this for ancestry reasons or for pharmaceutical research. A lot of the data that has been collected so far is, in fact, just SNP data, but they still have the samples. And I can only imagine that they're planning to do full genome sequencing as soon as that price hits an appropriate amount. Huge industry, tens of billions of dollars, and growing at a rate of 20% per year. So tons of data sets are being collected around the world. Different groups are collecting. Some of these are academic, profit, some of these are government, many of them are corporations. In the bottom left there, GEDmatch or GEDmatch, it represents a nonprofit that doesn't actually generate their own data, but rather collects data from individuals who intentionally submit the data, willingly submit the data to the platform. The range here is broad, and the potential for doing good is immense. But there's also groups that are just collecting as much data as possible and in ways that might not be entirely obvious to people. Because when you submit for a medical test, for example, that medical test may be sent to a large institution overseas that then sequences it and gives you the data, and they may keep the data for themselves and you never know about it. In fact, many times people willingly participate in research studies. You go to the doctor, okay, I'm gonna do this test, or do you wanna help research? Of course I wanna help research, but then your genetic data or your genetic material is now in a repository or in a data set. And as we'll see, there's really no going back from that. They tell you, however, that it's been anonymized, and we'll see here shortly that's not entirely accurate. And many people might not know that their data is already, or their DNA is already in a repository. So since the early 80s, the DNA of every baby in California is being stored. So they take a tiny little bit of blood from a child as soon as it's born, and then they put it into storage. Where is that going? Who has access to it? Has that been turned into data? These are all very important questions. Are you in the database? Well, if your DNA is in California, in that hospital, then there's a chance that it's already been converted into electronic data. And I don't know exactly how you could figure out where that information is going, but there has been some news articles examining the types of research that they're essentially selling this material for. So we have some protection around this, right? We hope so. There's the HIPAA, which protects all of our medical information, and that has some excellent, strong penalties for hospitals that willingly or knowingly violate this law. The law prohibits the sharing of personally identifiable health information. So we can't just openly say, okay, this is the name of the person and this is the diseases they have. But it also has this loophole where you can share information as long as you anonymize it. And their definition of anonymization is incredibly weak in that they simply require you to remove things like name and address and age from the data set. And data scientists are continuing to show more and more that it's very difficult to anonymize information, and simply removing the name is rarely sufficient. So the next thing that we have protecting us is the Genetic Information Non-Discrimination Act, and that prohibits insurance companies, for example, from charging you health insurance rates based on your genetics. It prohibits employers from discriminating against you based on your genetics. It does not prohibit life insurance companies from using genetic information to change your rates. Maybe that's reasonable. In fact, maybe it's even reasonable for health insurance to start including that in your costs. So we have this law that's supposed to protect us, but there's a chance that law could change in the future. So your DNA is out there, the law changes, well, then you're in some trouble. Furthermore, it's not clear exactly how you can enforce a law like this, because how do you know for certain that your employer or that your health insurance company is doing some type of discriminatory practice based on your DNA, or it could be told to you that it was something else, but in fact it was related to DNA, and so it certainly gets murky. Point here is that relying on laws to protect us is likely insufficient to really protect the entire scope of potential threats, as we'll see. And as I mentioned, the data can kind of get out there and go everywhere, but it's important to realize that these testing firms are sharing your data more than you think, some more than others. One could say that some startups may have their primary business model as essentially selling the genetic data. But that's not clear to you when you participate in the test. And another aspect that one must consider is that if you read the terms of services of pretty much everything, it says, well, we're going to have all these protections for you, but if our company or whatever gets bought by somebody else, it's all property of the business. So there's no control at that point. So there's no guarantee once you put your data in a database almost anywhere that data is going to be under some type of strict control. And should that data be breached or leaked, it's worse than any other type of data breach that's possible, because we can change our credit cards, of course. If you have some private information in your GPS and GPS data gets sold, maybe you can switch up your patterns. You could move. I mean, it's not easy to change some of these things, but it's possible. With your genetic data, it's effectively not possible to change your DNA, not in any way that will protect you in a genetic data breach. So once the data is out there, there's no going back. So you might think you know where your data is going, and then all of a sudden it ends up all over the world. And that could be because it's been shared, because your data was in a database that was then belonging to a research group, and the research groups pool all of their data together. So you may click that, okay, I want to participate in research button, and then all of a sudden, all of the researchers that are part of that program now get access to that data, and that, controlling security, information security when you're sending data to a bunch of academics all around the world is next to impossible. But this data is also very useful. So this first showed up, one of the most amazing forensic applications was in 2018 when the Golden State Killer was captured based on his genetics alone. And he was a serial killer from the 80s all over California. They had pieces of his DNA left at the crime scene, but he was a former police officer, so they had no idea who he was because he knew all of the different police tactics. But researchers took his DNA from the crime scene from the 80s and sequenced it and put that into GEDmatch, that database of people who essentially are looking to find their long-lost relatives. And they found his relatives. They found a few of his relatives and narrowed down the potential person based on, okay, he had to live in California during this time, et cetera. And then they followed him, and they actually physically got a piece of DNA from him himself and matched it one-to-one with the DNA that they found at the crime scene, and boom, they've caught their killer. And that's more or less with just DNA alone and a publicly available database. And this forensic technology is adapting. So for example, we have here Parabon Genomics that offers what's called Parabon Snapshot. And they are working on the ability to predict somebody's face and other traits based on DNA alone. So you can imagine some criminal leaves their DNA somewhere, and through their forensic capabilities, they're able to pull a picture of that criminal, and that might help catching the criminal. It's somewhat somewhat complicated to do this approach through machine learning. There are definitely improvements to be made. As far as I'm aware, only one person has been caught with this, and that's because they showed the composite image on the news, and the criminal saw his picture on the news, knew that they had his DNA, and turned himself in. Next slide. The technology is advancing. So Human Longevity Incorporated worked on full human genome sequencing. This is more data than just the SNPs that we've been using before. And they worked to reconstruct faces. And so you can see on the left is the real face, and on the right is the reconstruction. Again, somewhat controversial because they used multiple different ethnicities? Could it be that they're just pulling, ethnicity out and taking an average ethnic face? I suspect that this technology, as more data is produced and as better machine learning methods are available, that this approach will become better and better at predicting faces. And they also work to predict voice. So can they reconstruct a voice perfectly? No, but could they get aspects of your voice to perhaps rule suspects out in a criminal investigation? Entirely possible. So your DNA has all of this information about you. It has your ethnicity. You can estimate height, weight, hair color, eye color, last name if you want to look into databases. What the consequences of this are is that, of course, your DNA is not anonymous, right? So if you leave your DNA somewhere, someone might be able to figure out who you are. But importantly, it means that your genetic data in a database is not anonymized by simply removing the name. Because if you can take that data in a database, for example, compare it to data from the publicly available databases, if you have access to these large companies, then it would be relatively easy to find a large number of your relatives. It only takes a small percentage of a population, of 2% of the population, to be able to identify the majority of that population. So tens of millions of genetic tests, pretty much there with the data that has been created. It's not all publicly available, but essentially your genetic data in a database is not anonymized, which is somewhat of a problem for these the data sharing that we're doing today. And so these future advancements hold a lot of promise for us to be able to identify people, identify killers. Can we perhaps catch killers before they even start? Like in this Minority Report movie here, unlikely in our lifetime that we'll be able to pool all the information together in the world with genetics and solve crimes before they happen. But we're headed somewhat towards that direction. And so these future advancements all of a sudden become future threats. And it's not just about the serial killer that can be caught, but also about the undercover agent, perhaps, that's working deep undercover for years under an assumed name to infiltrate a criminal group. That individual is now at risk simply because we have not been cautious with the way that we've generated and shared our genomic information. This, of course, has serious implications for members of our military and intelligence community, because people working undercover have their lives at risk if their identity is uncovered. And special forces operators, for example, who go on operations overseas, there's a chance they leave DNA behind. And the last thing they want is for our adversaries to know exactly who did a particular raid that captured, say, Osama bin Laden. So how easy is it to identify people? Do you need to have this advanced machine learning technology? Not really. I joke that it's so easy that a reporter could do it. A BuzzFeed reporter took DNA from 10 of their colleagues and was able to identify all 10 of them. Then a researcher who was actually had experience in this technique only took 3 hours to identify a reporter based on her DNA alone. So relatively easy to do today already, meaning that if you have access to a database, you can look up how these reporters did it, and you can identify people in that database. And adversaries already know this. In the UK, the hospital system there has been warned that by the Ministry of Defense in the UK, that hackers are coming after genetic data. And these DNA sites have these issues around hacking. There's no clear evidence that any particular site has had their data hacked yet, but the hacks around these sites and on these sites that may or may not have taken the data should make people think twice about whether or not they should be sending their data to these sites. Because they really come after our data, right? We can think of here where hackers publish personal data on thousands of federal agents and officers. We perhaps in this room know about the OPM breach, where a lot of personal information was taken about our government officials. And with genetic data, once it gets out there, there's really no going back. You can change your patterns, you can change your address, but your genetic data, it's out there, it's out there. So we have this problem with unsecured genetic information. There's not a lot of attention paid to security, as we'll go into detail in a moment. The data is identifiable and really needs to be started to be considered as such. We can no longer simply remove the name from the file. There is certainly the potential for private information to be present in this data. For example, if you have inherited a neural degenerative disease, then that's right there in your identifiable genetic data. You might not be aware of that, and somebody who gets access to your data could potentially blackmail you with that or use that against you in some way. Relatively straightforward, doesn't affect everybody, but it's a significant threat. And so how long, or are we already seeing perhaps that the black market is starting to collect and sell this data at some rate? Now we start getting into biotechnology and start considering that genes can be edited through CRISPR technology. Is what we're using today, is what it's called. And there's been a warning out for years now from James Clapper, who said that it's a weapons of mass destruction threat. So you can turn biology into a weapon. You can modify the virus, you can modify the bacteria and make it something that could spread and kill a large number of people, or could just be a bacteria that makes a small group of people violently ill for a short period of time. Very recently, Congressman Jason Crow has warned that, quoting here, there are now weapons under development and developed that are designed to target specific people. That's what this is, where you can actually take someone's DNA and you can target a biological weapon that will kill that person or take them off the battlefield to make them inoperable. So this is no longer speculation by scientists that say that DNA can be used to build targeted weapons. This is something that apparently one of our representatives here has said have been developed. And we're waiting on some information to come out to Congress this year. It was supposed to be in June that they received a report from the Defense Department discussing this. It's possible that that report will provide evidence to back up this claim. So what kind of threats might we be facing? And I'm going to refer to great scientist, Dr. John Sotos, who at the time was the chief medical officer at Intel. And he discussed how DNA could be used to cause harm to individuals and populations. And I'm doing this because I recommend that you watch this talk, first of all, but it's very scary, so don't watch it at night before bed. And I also want to show that I'm not the only one talking about these specific threats. So there's a history of bioweapons, and that history goes all the way back hundreds of years ago when smallpox was used as a weapon. In fact, before that, the Middle Ages, where they would do things like chuck rotten bodies over walls when they were sieging castles. But modern history, smallpox, okay, anthrax, a nasty weapon. It doesn't spread, so it kind of makes it useful as a weapon. You don't have to worry about making your own population sick from it. Not trivial to build, but it's certainly a biological weapon that has some history. And influenza, and you might not think of influenza as a potential weapon, and it's never been intentionally used to my knowledge as a weapon, but we see this interesting history with the dominant strains of influenza in the population. So 1917, there was the horrible influenza outbreak, which killed millions of people and is known as this terrible flu disease. And that strain stayed around and evolved until 1957, and then H2 becomes the dominant strain, completely different strain. H3 shows up, again, completely different strain. And all of a sudden in 1977, H1 shows back up again, looking exactly like it did decades before. So the idea is essentially that somebody was keeping around that nasty strain of influenza, and it escaped from the lab and it spread around the world. Thankfully, While it made many healthy people sick, it was not anywhere near as bad as the 1917 pandemic. So there's a lot of potential threats out there. Smallpox, when we look at things like the Zika virus and how it causes serious developmental issues, there are diseases that exist that are incredibly frightening. And so what's stopping people from developing these as weapons? We have a bioweapons treaty, and as some of you may know, it is horribly underfunded at the UN. There's not enough money going to this. There's not enough attention going to this. So while we have the treaty, it's just, there's really no way to enforce it. Technology is still being developed. As I mentioned, we've got CRISPR, but all of this technology is not yet in the hands of the average scientist. That's rapidly changing. A major thing that may be stopping bioweapons is the potential for blowback. That 1917 flu in the lab, that could escape and that could cause your population sick. And the person who's operating that lab himself might be at risk from that disease. One idea is that genetics allow you to target a disease towards a certain population or towards a certain individual, perhaps reducing the effect of blowback. So the blowback that may occur to your population could be low enough that you might not care too much about it, because you know that the effect of just making the other army sick, for example, could be strong enough. So before the pandemic, Bill Gates said, of all the things that could kill more than 10 million people around the world, the most likely is an epidemic stemming from either natural causes or bioterrorism. An epidemic is one of the few catastrophes that could set the world back drastically in the next few decades. So we saw a taste of this with COVID-19, and unfortunately, the concern is that that is inspiring more people to seek the development of these weapons, and genetic information is potentially a key component of that. So in the future, we can start to think about what might happen. And there's a range of different ways in which bioweapons can be used, for example, against species. There's people working today to try to rid the entire world of a specific type of mosquito that transmits the malaria parasite. That would be a wonderful thing. But at the same time, we rely on some species of animals and crops that have very limited genetic diversity, which means that perhaps they might be relatively easy to wipe out with a bioweapon. And that would affect our agriculture, perhaps our bioeconomy. When we start thinking about how problems can be made for groups, this leads towards people who may want to attack entire ethnicities. And it can get all the way down into the individual, where they could target an individual with a genetic weapon just for assassination or to debilitate them. And this can range everywhere from being an inconvenience, maybe they just make you sick at the right time, so you're unable to give that speech, or could be something that's worse than death. And Sotos talks a little bit more about this. Essentially, we don't want to go there. We don't really want to know. What we want to do is we want to work really hard to prevent these types of things from happening. And so, as we've seen, next slide. As we've seen, we can use genetic information to uncover our ancestry. But the consequence of that is today we have lost our anonymity. And that's essentially already happened because of the way that we've treated our genetic information. Also, in the future, genetic information will be used by your doctor to predict which diseases you may be predisposed to and provide you with specific treatments tailored to your genetics. This is called personalized medicine, and it holds the hope for great advancements. Advancements that we need desperately in medicine. If anybody knows things about pharmacology, that some people mysteriously have horrible reactions to some drugs, and other people get their disease cured by it. So we certainly need to make these advancements, but at the same time, that genetic information holds the information about your illness or your potential illness. And there may be things in your DNA information in your DNA and your genetic information that you don't fully understand, that you don't know about, and that could be used against you. Things related to mental illness that you may be predisposed to, things related to neurodegenerative disease. And so just like genetic information can be used in medicine to heal, it can also be used for harm. And when we look at amazing biotechnology advancements, CRISPR, for example, these things lead to awesome aspects of the bioeconomy and cures for diseases. But as this technology develops, it can be co-opted to be used for bioweapons. And a challenge that I see in the biotech space is that it's very difficult to know exactly what's used, what can be used for good and harm. So we move forward with biotechnology as quickly as we can, and then it seems like that potential for harm is an afterthought. And we let the cat out of the bag and we create a machine perhaps that can automatically build your organism using CRISPR. We're not there yet, but imagine that, you know, that's so cool. We can design that yeast strain to produce whatever chemical we want. That same technology could potentially be used for harm. And how do you keep the lid on that? So genetic information is at the heart of that. We need to protect genetic information because that is one way to mitigate these threats. If biotechnology is running rampant, well, at least if we protected the genetic information, right? Unfortunately, today, that's not the case. So for example, the most common DNA sequencing platform is the Illumina sequencing platform. And just recently, the FDA and Department of Homeland Security have warned about a particular vulnerability that scored a 10 out of 10. This vulnerability allowed hackers to gain full control of the device, obviously steal the data, potentially modify the data, and of course they could knock out the sequencer if they wanted. They could shut it down and turn it into a very expensive brick. We're also seeing very sophisticated hackers building malware that's targeted towards the bioeconomy, biomanufacturers in the laboratory. The tardigrade is this image here. It's a very small organism, also known as the water bear. It's amazing. It can survive without air and water for decades. It even can survive in space. And they've named this malware that's infecting our laboratory after the water bear. So sort of indicates that you've got something in your system that survives without air and water. You know, how are you gonna clean that out of your system? And even if you do, even if you're able to somehow go through every single piece of electronic equipment in the lab and clear it of this malware, you've got a highly capable adversary who's actively coming after you. So could they make tardigrade number 2? Of course. And then the question is, what else is out there that's like tardigrade that we have yet to find? Point being, the devices aren't really secure, and we've got serious people coming after these devices. So how do we generate genetic information in the lab? First, it starts as that DNA sample. That is the molecules that's then obtained by a lab tech. We absolutely need somebody in the lab. No matter how much we automate things, we have to have somebody there to at least look over the machines. That introduces the human element. People make mistakes. People accidentally click on links, this sort of thing. And then we need to generate genetic information with a DNA sequencing machine, right? That's how we want to be doing this in the future so that we can capture all the variation that we saw in our first 2 slides. And if those machines are hacked, then that information can be stolen as soon as it's produced. But from there, that information then has to go to a lab computer. The lab tech generally needs to look at that information and say, okay, experiment worked, good, I don't have to run it again. Send that information off, hospital, cloud, wherever it needs to go. And so this is what we'll refer to as a kill chain, where along the line here, there's a number of different ways in which that information can be stolen. So how do we protect that information? The work that we're doing with CRDF Global is to provide security guidelines for laboratories in sub-Saharan Africa to help them improve their cybersecurity. And for that, we recommend things like data access control. You can't just put the data, on the web with an open link. You need to have some control over who has access to it. Well, how do you do that well? You need good passwords. You certainly should have multi-factor authentication. So it's not just a password, but other ways that you can verify that the individual accessing the data is who you want to access the data. And then from there, you also need to review the logs to say, okay, well, that person works during the daytime and all of a sudden at 3 a.m. their password has been used. Okay, let's look at that. Let's make sure that that was them. All right, you can detect a breach there. You have to constantly be paying attention to these things. Simple access control, but we still have the human element. So people make mistakes. People can be socially engineered. You could leave USB sticks lying around in a parking lot and make them really cute. So people pick them up and think, oh, that's really great. And they go plug them in the computer and the network has been compromised. The insider threat is not just an individual that intentionally does something or is paid to do something. They can accidentally do things to compromise your systems. So, so how secure are our biological laboratories? I hinted towards some of the research that we've done, and next slide, we'll see that we've sent a team to find out, but we'll first talk about what labs need to worry about. And the first thing they need to worry about, at least in my opinion, is confidentiality. We need to protect the data. So as a major factor of these labs, they have to focus on making sure that that data has remained in their control. The second most important thing, especially after the pandemic, is the availability of the lab. Not just the data, but the availability of the lab, because what we've seen in COVID is that the laboratory infrastructure is critical infrastructure. Without the laboratory infrastructure, we wouldn't be able to detect where COVID was, sequence the different strains, determine which people are ill. So this critical infrastructure needs to be maintained so that it is available when we need it, so that it's not slowed down or stopped. And hey, that experiment didn't work. You got to run it twice. There's nefarious ways to reduce availability in a lab that's not entirely clear that you're being under attack. Finally, the integrity of the system is important because you want to be able to trust your data. Not trivial to go in there and modify data on the fly as it's being created, but it's a possibility and something to consider. So what are the costs of this insecurity and who's paying for it? Well, of course, if genetic data gets breached, there is a cost to an institution's reputation, but as we all know, a good PR marketing team can overcome that. The potential for intellectual property loss may be more compelling in that in the bioeconomy of the future, almost the entirety of the intellectual property is in the DNA of that organism that you've designed. So you've spent billions of dollars to make that bacterial cell that can do some magical thing or create some amazing chemical that was difficult to create otherwise. And if somebody is able to steal the genetic information, then they can reproduce that organism almost perfectly. And now they have exactly the technology you spent billions of dollars on. So a little more compelling. There's, of course, regulation around this. I mentioned the regulation that we have that covers us nationally here in the United States. There's also CCPA, which is California, that does call out specifically genetic information. In my opinion, it needs to go further to discuss identifiability of data and things. GDPR in Europe is the strongest of all of them. It's still not perfect, but it does have some strict penalties for violations. And all of these can have a cost, which again compels people to perhaps pay attention to their information security. But what we're looking at here is that our bioeconomy and our biosecurity and our health security, they're all at risk when this information is lost. So it's more than just a cost to an institution. It's really a cost to entire nations. It's a cost to ethnicities. It's a cost to families. And it really depends on who's being targeted, who the adversary is, and what type of data has been collected. So this goes beyond an institutional risk and really becomes a national risk that we all need to pay attention to. But this risk, this cost is not just on us as individuals or on our families, but can in fact last for generations. Because the genetic data is passed down from one generation to the next, that means that the costs to our future generations could be high depending on how we decide to treat our genetic information. So we sent a team to find out how different labs handle laboratory cybersecurity. Working with the University of Colorado, we performed data security assessments on different labs, academic labs, nonprofit, for-profit lab, and we examined the IT security in these places, looking at their networks for vulnerabilities, looking at how they handled their data, and found that insecurity was essentially the state of the art. We've published this, next slide, in a Frontiers journal. We found that, passwords were weak. They had them on sticky notes, these types of things. The physical access to facilities matters a lot, right? If you're in an academic institution and anybody can just walk in and out of the lab, well, that becomes an issue. And insecure devices were found, and this is essentially key to our understanding here, in that devices are just not secure. It's hard to make secure devices. Apple works hard at it and constantly needs to update, and comes out with new chips and new devices, and they still have to struggle with it. So so we can see then that device security relies on the device manufacturers. You can't have that expert that can reprogram that device necessarily as an institution. And in fact, many of these devices, you're not even allowed to go into their system. You're not allowed to take the panel off. It voids the warranty, and you can never use the device with that company again. So you have a security issue. You have to call up the manufacturer. Okay, I found this. Okay, and you work with them on that. It's not something you can fix overnight necessarily. Some devices are just impossible to secure, and you don't need them in the lab. So simply you could remove them, but they're cool technology. So we want to have the coolest, neatest thing in the lab that does this neat thing, like play music or cleans our floors, as we'll see. But some of our devices in the laboratory are absolutely necessary. We can't remove them, and therefore we have a serious challenge when we see some persistent threat actors coming after these laboratories, because these devices are just internet connected, require internet connectivity, and not built for security. So how do we protect IT infrastructure if these devices are insecure? It's a major challenge. One recommendation is to go old school and to sweep your own floor and not use a Roomba. And okay, that Bluetooth IoT speaker, I don't know any security issues with it, but do you need it, right? So there's no known vulnerabilities with that. But if you have the boombox, you know that nobody's going to hack it. So the Roomba is an interesting example, because just very recently, so Roomba has a camera on it, and it maps out your entire floor plan. And just recently, you might say, oh, well, that's okay because they're not selling the data and they're keeping it secure. But recently the company was bought. So you never know where the data goes, right? That's a major theme of this talk. The more advanced the device, the harder it is to secure. These devices get incredibly complicated. They have lots of different electronic equipment in them. Some of them run on operating systems, but even when they run on operating systems, they still have software that's embedded into the device that is required to run the device. I mentioned physical access to laboratories. Well, if you've got USB ports on the front of your device and a DNA sequencer right at the front of your lab, at the front of a door that's not really properly secured, then it's relatively easy for somebody, either intentionally or unintentionally, to maybe go in and use a USB stick on that device. You could potentially glue those USB ports closed, but it's a very expensive device. You might want to resell it, and is that going to affect the resale capability? These device vulnerabilities can take a long time to be addressed by the manufacturer. The bigger the manufacturer, the more they have to lose. But a lot of these devices, like say DNA sequencers, are not necessarily built by big companies. There's startups that come out and build the next greatest technology. Hey, look, we've just dropped your costs tenfold. It's only $10 a a genome, to sequence your entire genome. But that device has been built overseas. So now we're going to need to address that. Somehow we're going to have to put that device into our, into our network, into our laboratory, and work with that device, because we need that information. We want to save money, but we can essentially throw device security out the window. So we have a revolutionary solution to this: molecular cryptography. This is the application of cryptography directly to DNA molecules themselves. This protects genetic information before it ever touches a computer. So that issue with that DNA sequencer being insecure, or maybe in a lot of small countries, they don't have the capability to do DNA sequencing themselves at all. They have to send it overseas. This helps to resolve those issues. Essentially, it focuses on what we're going to refer to here as security tags. These are small pieces of DNA that can be used to tag other pieces of DNA, physically link them together. DNA has A, T, C, and G. And so with a DNA molecule that is, say, 30, 40 base pairs in length, let's take between 30 and 40, there's a very large number of random molecules that can be created, right? So we could essentially tag every single molecule in the known universe with a piece of DNA, with a unique piece of DNA. So that's 10 to the 33 or something like that. Numbers that are just entirely just crazy big. So we're tagging pieces of DNA. So we take samples and we tag each piece of DNA in that sample with a unique tag. So each piece of DNA in each sample gets a unique tag. Now, when we pool those samples together, next slide, we get just a jumble of DNA, just a big mess of DNA, and each piece of DNA has its own tag. Only if you know which tag belongs to which sample can you then decrypt the data. So just by pooling samples together and giving them unique tags, we've effectively anonymized the information. The information that allows you to identify an individual is more than just a tiny piece. You need information from across the genome. Say, okay, we're sharing information with this relative over here and this relative over here. We think we figured out who this is. You can't do that with a tiny piece of DNA. Furthermore, what we can do, we can take more advanced methods where we start intentionally adding things into the pool to add decoys, as we would call them. You add decoys to the pool and you increase obfuscation. So if we were, say, targeting a single gene in a large number of people and adding a large number of decoys, you get excellent information obfuscation to the point that if somebody has access to that data, they really gain zero information about it. This is not a major change in the way that we do genetics. Today, we are already tagging samples with pieces of DNA, with sample tags, sample index tags. And the issue, though, is that each sample gets its own tag. So every piece of DNA in sample A is one tag, every DNA in sample B is another tag. So we're tagging and pooling DNA to save costs on the DNA sequencing. We can do it in mass. But the data that then comes off of the laboratory computer, well, it's trivial to figure out which piece of DNA belongs to which sample. So you've just done whole genome sequencing and you have created this information. Somebody gets access to that. They have the entire genome. They can identify it. They can potentially use it against you. In contrast, our approach here is still tagging samples, but because we have unique secret tags for each piece of DNA, then the information is secured before it ever touches a computer and becomes anonymized, and the health information is effectively concealed depending on which application you're using. So you can imagine, okay, well, we need to trust these tags, right? We need to trust GeneInfoSec perhaps to build these tags, and now GeneInfoSec holds the keys for us, but that's actually not the case. We're building the technology so that institutions that use our approach have complete control over the keys that they need to decrypt the data. We do this by chemically generating random security tags. So we create a pool of a large number of tags, but we have no idea what is in that pool because it was generated with random chemistry. And again, 10 to the 33 is a very large number, so we can create randomly a very large pool of pieces of DNA and yet still have no idea what sequences are present. But then the groups that want to use this technology can sequence those tags in a way that allows them to still use them. And so that allows them to receive a pool of random security tags and ensure that only they know which sequences are present in that pool, allowing them to have complete control over who could have access to the data. Now, when we get back to that kill chain, we can see that this entire process of data generation has been secured. Nobody needs access to the sensitive information, but the lab tech does in fact need some access to the data. So the lab tech does have to look at that lab computer and does have to say, oh yeah, okay, that looks like my sequencing run worked. Maybe they didn't get every sample in that run perfectly, but at least they can confirm that the data has been generated after they produced it. On that lab computer, that's still running Windows 7 and nobody wants to update it or anything like that, right? This is how a lot of labs work. And then from there, it can go onto the cloud, right? Where the data is then secured on the cloud, then secured the whole way, and you can have access control to it, right? You want to pull down a lot of whole genomes, maybe you want to take that analysis to an offline computer, something like that, then decrypt it there. So this avoids backdoors being built into sequencers overseas. It reduces the amount of lab security required. Of course, we need to pay attention to lab cybersecurity to prevent them from being taken offline and this type of thing, but it's a lot harder to detect data being stolen than it is to detect a lab being shut down. And this does provide an entirely new form of data security. Not discussing it here today, but DNA actually can be used as a data storage medium. So we can store data in DNA and it lasts a long time and is stored in a small compact form. And so our technology has applications there as well. And so I put together a great team. We have a core team with expertise ranging from cybersecurity to business to genetics. And our team of advisors has as well a range of expertise from software development to leadership to information security and of course biosecurity. So with that, I believe we're going to open it up to some questions.
Hi, everyone. I'm Dr. Julie Fisher. I'm the technical director for global health here at CRDF Global, and I have the pleasure today of moderating some of the Q&A and the discussion period here. I think before we launch, while we are waiting for more responses from you and your questions, which we'd ask you to please input into the chat box, we have a couple of polling questions, one to get started with to invite everyone who is participating completely online to share some of their own thoughts. Coming soon.
What's up?
Okay, great. I think Barbara will be relying on you to read us the results because we can't see them.
Yes, we have one more minute to submit responses.
And again, while you're responding, I would like to encourage everyone online to please go ahead and submit your questions, and we are compiling them and we'll be going through them in just a moment.
So we have our results. If you are viewing this remotely, you should be able to see them. So majority of our online viewers are from the private sector and academia. They close second to other.
Great, thank you. It's always great to hear who's out there. So our choices were government, academia, private sector, and other.
Okay.
Great, thank you. So I think we'll go ahead and start with some of the questions. One of the questions that was posed was that you're talking in some cases very generally about genetic information, but genetic information varies widely and it's used for a lot of purposes, including in addition to next generation sequencing to look for variants in the human genome for specific diseases. There's tumor characterization, there's pathogen identification. And in some of those cases, we're actually actively promoting sharing of that data more effectively. So one of the questions is, how do some of the recommendations about being more conscious about security of genomic and genetic information generally align with frameworks that are being developed globally to promote responsible sharing of genetic data, which again, genetic data comes in a whole lot of different varieties of flavors.
That's an excellent question and something that the scientific community very much needs to be discussing and addressing. As you mentioned, there's a range of different types of genetic data. So to begin with, we have to, I think, start by asking which of this data is easily identifiable and which of it is more difficult to identify. From there, we then need to begin to consider how sensitive that information is were it to be identified. So I would never assume that the information is impossible to identify, but we may say whole genome data, much easier to identify. So we're going to put that in a different category than say agricultural data, which it doesn't matter to identify. Sharing that information is critical to building these large data sets and doing open science. And open science is conflicting with the work that we're doing, because open science is how I did my PhD, right? I mean, I got a lot of data from other scientists. It wasn't completely open necessarily, but I got data and did bioinformatics on that data. And I really like the idea, as most scientists do. But when we have the capability of causing harm to our participants, we have to be very cautious how we proceed. So there is no easy answer. One key point after the discussion has been made is of how, what's sensitive and what is less sensitive, is how do we then keep it secure? Because today, perhaps we simply require researchers to check a box to say that they're doing certain security protocols. Can we improve that by actually having them go through training? These are little steps, but we need to move forward with these little steps to get to the place where we need to be, where the most sensitive information is kept protected with the greatest security.
Thank you very much. And I think that leads actually to some of the questions that a lot of the participants have about the risks. So you have spoken about some of the potential risks of individualized or targeted biological weapons development based on these sequences. Some of our participants have asked about what you think, if you could present those risks kind of in a continuum, what are your concerns about the risks of stolen genomic data? You've talked about the individual privacy issue, but other risks.
As in to nations and.
Yeah, I think on the larger geopolitical scale.
Yeah, again, another thing that was learned from COVID is that the bioweapon attack is not just something that kills everybody, right? It can be something that shuts economies down. It can be something that causes troops to be knocked off the battlefield. And that can spread very quickly as well. So the Omicron variant was just a super spreading virus that there was really almost no stopping. So with that in mind, some of these risks pertain to more subtle negative effects that bioweapons can have, not just assassinating individuals or trying to wipe out ethnicity. so at the national level, at the national security level, that can be seen. But as we sort of zoom in at the ethnic level, that can also be a concern, because maybe the next whatever contagious virus comes out there is going to be that much worse for a particular ethnicity. And if we start getting into developmental genetics, it gets into that scary territory that we have to worry about it. One major issue that we must consider is the timeline, right? So how is this going to evolve? Well, 40 years from now, the threats that we'll be facing will seem like pure sci-fi today. So we can speculate about them now, but it's all we know is that it's moving in that direction, and then that there's becoming greater and greater danger in this area. So a kid born today, if their DNA is put into some database to solve childhood diseases, I'm a fan of that. But if we just put that data out on the internet where we can get access to, there's a good chance it'll still be around when that kid is 60, 78. So when that kid is in a key leadership position and also frail and vulnerable. So whatever threats we may be facing in the near future, we can say for certain that things have the potential to get incredibly serious in the more distant future. And it's for that reason that we need to actively start protecting genetic information, especially of younger people.
And I think a follow-on question from one of our participants is very related to that. There's already an enormous amount of information available at GeneBank and other storage sites, which is there specifically to assist in research and to improve our ability actually to sequence. And it's key to understanding better how to use raw reads, particularly as the new platforms generate a ton of data that can be difficult, particularly in laboratory environments where researchers and public health and clinical laboratorians are just really beginning to understand how they could harness this technology for benefits. So what do we do with that vast amount of data? You're already saying, you're talking about individual protections for genetic data that might be available on the internet. This is data that's intentionally stored in large and accessible databases that are used primarily by scientists for to improve our understanding of causes of disease and improve our ability to diagnose and treat those in the long term. So what do we do with that vast amount of data? Are you concerned about what's already stored in GeneBank and in other public international databases?
There, again, open science issue where we want science to be open. Those databases are incredibly useful. The data is already out there. Personally, I wouldn't want to be in one of those databases. Maybe after this talk, you might agree with me. But nevertheless, the data is useful. Just like with molecular cryptography, you mentioned the need for sequencing platforms and this type of thing. The technology that I proposed here today helps to resolve that specific issue, in that we can put on sequence data of large amounts of data that has better anonymity to the data, right? We could put a sequence of decoy data, of data that didn't come from a person, that was synthetically generated, so that we can say, okay, this is what the sequence should look like of that synthetically generated piece. It doesn't have to be that, well, it's complicated, so we're just gonna keep putting full human genome sequences out for the public to access. That's ultimately going to be more problematic for scientists in the future, because as people become more and more concerned, so an example is Henrietta Lacks was a person who ended up having her cancer cells being donated for her to science, and it's used all around the world by scientists. Most people today would not offer their cells to become immortal for all scientists to use. And in the same way, that genetic data being out there on the internet may cause people to be more wary and more cautious about how they're willing to share their genetic information, especially as we see these threats around targeted bioweapons emerging. So if we as scientists sit back and say it's complicated and we need the data and we're going to do nothing, then you're just going to hit a point that says, okay, now we absolutely have to do something, and the public is unhappy with the way that we've been treating the data so far. Not an easy answer, but there are solutions, and until we recognize the problem, we can't start working towards
I think some of our participants would like to back out a step. And given what you've said about assessments of laboratories to understand the general risks of data, the lack of data protections and the need for cybersecurity. We have a couple of related questions about, given your observations globally, what would you recommend or what solutions can you propose for laboratories that want to consider and assess their cybersecurity risks? So if you could talk a little bit about tools and approaches. And then a second related question is what is the right balance between hardening laboratories against developing the cybersecurity tools to harden laboratories against deliberate theft at the laboratory level, and then looking more broadly at regulations and policies?
Okay, multiple questions
Yeah,
there.
two questions.
Two questions. So we'll start with what do we do, right? Okay, we're running a laboratory. What do we do to address this? I will say that many more recommendations and guidelines are coming out. So the need for specific guided details, that's in development, right? Especially with CRDF and a lot of good work everyone's doing here. So specifics on that, we'll say, okay, pay attention and wait for those laboratory specific guidelines to come out. There's nothing stopping a laboratory from doing standard cybersecurity practices. So for example, in the United States, we have things like the NIST cybersecurity frameworks that I recommend, or at the international level, there's also international guidelines that can be looked up, and that would be the ISO framework is a good one that we recommend. And you just go through the list and you say, okay, this is all the things that I have to do to keep my cybersecurity, keep my data secure, keep my laboratory secure. But really, that can be overwhelming, especially to a laboratory that doesn't have a dedicated IT staff, let alone a dedicated cybersecurity expert. So start small and work towards each change incrementally. Get that, as I mentioned in this talk, critically, get that access control and create logs and review those logs. Don't wait 2 years to say, oh yeah, somebody's been in our system for 2 years and we just checked it out and it's been a problem. No, you have to actively review the logs, see who's logging in, where the data is going. It's not going to solve most of the problems, but at least you've raised the bar a little bit, right? The second step that I generally recommend is get rid of those devices you don't need because you're just complicating your security posture by having unnecessary devices, and then use firewalls, and even better, use segmented networks. So, hey, I need to have this device over here. I don't think it's secure. Okay, put that on a separate network than the other devices. These are small steps that can be found in both the NIST and the ISO framework that can be worked towards to improve cybersecurity, but you've got a very serious, let's say theoretically, you've got a very serious adversary coming after you. Is that going to be enough? At least you've made them work a little bit harder for their meal, but unlikely to be sufficient. And so at a certain level, you have to say, okay, at what point is the data so sensitive that I'm going to need to take it offline entirely? And that is a challenge, but is a potential easy solution to say, all right, just take everything offline and work as much as you can offline. And if you could remind me of the second.
How to complement the leadership from policymakers to develop regulations and policies to address the concerns at a larger than laboratory level.
This is an incredibly complicated situation because lawmakers are typically not experts in science. So, so how do we get it? First of all, we need to get the experts to talk with the lawmakers. That's first and foremost. They need to be informed of what's going on. Then policymakers and lawmakers really need to consider the landscape and consider the time scale. As I mentioned, we're talking about decades of potential threats coming in. Consider their particular country or institution. What kind of threats are they facing today in this area? What kind of threats will they be facing in the future? And then act accordingly. There is no one size fits all here. There could be some laboratories or some governance that surrounds genetic information from academic research on, yeast strain that everybody's already studied, and you can say, oh, good, clearly there's no threat there. When it comes to the genetic information of a population, personally, I see these threats as being very serious and that we have to consider them as so and start, again, moving in that direction of at least asking, what are we going to do about it? And how do we actively implement those changes so that they can be, so that they can be actually enforced?
Thank you. One of the, we have questions along a whole different set of spectra here, but one of them, one of our attendees are really curious about what is the record of this? So what is the prevalence, do you think? And I know that this is a guesstimate at best, but the prevalence of attacks to test the cybersecurity laboratories or to even given widespread openness, what is the prevalence of cyber attacks on individual laboratories?
Here, I'm going to plug the BioISAC, of which we are a member. It's information sharing coalition.
Can you explain a little bit what that is?
Yeah, it's a group of companies that get together and share cyber information. So relatively new. I encourage people to check it out. BIO-ISAC, and we're working on that. It's new, right? It's incredibly new. It's an area that, because of the lack of attention, we've had a few years to start looking into this as a group of biologists / cybersecurity experts. So your question of how prevalent are these attacks? Water bear, the tardigrade, it's prevalent, right? And by seeing something that is clearly so sophisticated, we can say for certain that sophisticated people are acting in this area. Therefore, I would say that you have all these devices connected to the internet, there's a good chance that something is either going to be coming after you or is already there. How do we know? I'm just speculating, right? We haven't gone to all the labs. We haven't required labs to even have passwords, right? There's no regulation here. So I can only imagine from what we've seen in our somewhat limited sampling that this is just standard, that there's very few people who are taking this seriously. Now, there are certainly laboratories that take this seriously because there's some serious laboratories out there. I'm not saying all biological laboratories are insecure or all these companies have been hacked. I'm not saying anything like that at all. But what I am saying is that generally these laboratories are not paying close attention to security. The devices are really not very secure, which says to me that if they were serious about security, then the device manufacturers would have been contacted many, many years ago and would have been working to build security into their products. And there are good laboratories out there, but their task of keeping their infrastructure secure is quite formidable.
Thank you. Thank you. I think we're going to be starting another poll for our online participants, but we're just going to go ahead. If you're online, we trust your ability to do two things at once, so please respond to the poll. And we're going to go ahead and move forward because there are a lot of questions that are stacking up here. There's some interest here as people are responding to this general poll by people who are both familiar with sequencing and I think have some familiarity with encryption. The questions are related to the tags that you described in your proprietary approach. Specifically, questions about basically how you secure the tag pool. There's two related questions, I think. First, if there's a risk in having a known solution or range of solutions based on the individual pool, based on essentially how many tags you have to make that solution set intractable to decryption, which would be for people who are obviously a very determined threat actor. And then the second question is, does the specific tag remain with the data in perpetuity? So going back to the first question, if someone did crack it and make that public, would that mean that the data, all of that specific data would always be cracked?
Okay, so I I'm going to start with the second question. The first one is not entirely clear to me. I'm going to need a little bit of clarification. The second question is cracking. So this is not like other forms of encryption. It's essentially what's called a one-time pad. It says if you have this data table, then you can access this data. Without that data table, it's quantum resistant, I'll say, but essentially you're looking at very large numbers of random things that cause you to be able to, say this belongs to there. It's not crackable in the sense that we think of encryption as being crackable today. Of course, if your system is hacked and your keys are stolen, of course your data can be stolen. So it's just like any other system, you need to protect the keys. But I'll point out, this is an additional layer of security that is entirely friendly to every other form of data security. So there's no clash between using this system and using other systems. And then furthermore, this is a unique approach because it protects the information before it ever touches a computer. So you add that layer, and then you can add your additional layers of encryption on top of that, and then you can protect all of those keys, and you do the best you do to protect your information. Nothing is going to be foolproof. There's always a human involved, but if you keep the keys offline and you only decrypt things when the data is on an offline system, then you could have a fair bit of confidence that that information is physically impossible to leak out. Now, your first question was about the actual generation of the keys. There's no reason why you need to use a particular length. You could use randomly a large range of lengths. There's no cost to, there's a minor cost to using a larger length. I'm not certain if that answers the question. There isn't a, if you're using a range of lengths, there's no real length to be weakened, I guess. And so maybe you can go back to that question, see if I'm answering it. I'm not sure if it was entirely clear.
I think our attendee may want to follow up with a more specific question, but I think that what really what they were looking for is that essentially this question of once, if the tags have been cracked, I think is potentially the problematic word here, but if you're using a set of tags that repeat and they are associated with the data, if the specific tags would basically, like essentially if someone could use these and decrypt the tag, if that would essentially give them the key to all of the data associated with that for perpetuity.
Okay, so first of all, again, with keys, if they get the whole table, then they have all access to your, the full keys to your data. But knowing information about some tags doesn't give you the full table about what, you just randomly created a bunch of pieces of DNA, and you might be able to somehow say, okay, I know that these 5 pieces are going to be in there. That does not give you access to what those other pieces of DNA are. So, small amounts of what I'll call information leakage are theoretically possible, though unlikely. And that information leakage does not, unlike with traditional disk encryption, does not cause the whole system to collapse. Essentially.
Yeah. I think that's what they were asking. Yeah, thank you. All right, I think there's a mix of questions popping up. I think one of the questions that has been asked broadly, and it comes back to the question was asked before about how you balance the legal regulatory framework against what you can do at the individual lab level. But one of the questions is, what, in terms of legislation, are there any good models out there? Is there anything being developed that countries should be looking at, or that advocates for enhanced biosecurity should be looking at on their own?
Okay, excellent question. I'm going to point out that I'm a scientist that is building technology and not a legal expert. So I'm not here to say this is exactly what needs to be written into law. As I mentioned before, we need to get scientists and lawmakers to talk. And I think we also need security people in the room to say, both on the cyber side and the bioweapon side, to say, okay, these are the threats that we're facing. But let's consider who has actually been passing laws on this. And we'll start in, I'm going to say 2007, 2008, when Russia forbade any biological samples from humans from leaving the country. So, okay, somebody took action on this, right? They didn't just say, we need to protect this information. They said, nobody is letting their DNA out of the country. And specifically, the threat was a bioweapons threat targeting Russia. So they, that's been out there for over a decade now. More recently, China has passed laws on this that says that there are strict controls over who and who cannot be researched genetics, whose data can be collected and created in their country. So neither of those solutions are really great, right? And one of them you say, well, we're not going to send the physical samples out. And to my knowledge, they're not creating large databases. So they've kind of solved that, but they can't proceed forward with technology in this area without solving these other cybersecurity problems. And China's kind of solving that in their own right in a similar way of saying, well, we're going to at least protect some of our citizens from being put into these databases. This is my interpretation of their law, at least from 2018. And these are pretty strict policies if you think about it. They can hamstring the way that businesses operate. They can hamstring the way that research is done. We generally see scientists, maybe we'll see some more questions on this, but pushback from scientists who say, wait, we're sharing all of this genetic data and we need to keep doing this because if I don't have a data set that's this large, I won't be able to cure this disease. And I have to cure this disease because I'm really passionate about it, or it's personal to me. And there's the lawmakers have to address that with these trade-offs. Do we need to be as extreme as not letting genetic material out of the country? Maybe we should question where it's going.
I mean, there's a bit of an economic aspect to that too, right? I mean, you've been focusing more on hard security threats and weaponization, but certainly there's an enormous economic aspect to the use of genomic data. And you've already alluded a little bit to the idea of using these large data sets for essentially, for developing and testing algorithms. So in addition to some of you talked about this being decades out problems with some of the biological threats, what about the economy of these large data sets and genomic data that is available?
Well, many of these large data sets have been estimated to be worth a large amount of money. So there is an economy there that says, okay, this data has potential value. Has it been turned into actual value yet? We have yet to see it. So companies are pursuing genetic specific pharmaceuticals or treatments. Those companies have the potential to cure very specific diseases and improve medicine. And I'm all for that. I'm excited about that. That's why I became a geneticist to begin with. My interest is in medical genetics. But we are harming our future research by treating the data with negligence today. Because if we today say, well, there's an economic aspect of it, so we want to push everyone who wants to talk about security to the side and move forward making a lot of money, when you've got tens of millions of people whose genetic data is on the black market, where do we go from there, right? And then how hard is it to get people to say, yes, I want to participate in that research project? Not only that, but already we have biases in these data sets. It's a bunch of white males, or rich white people, essentially. That's a problem for personalized medicine being able to be of value to other ethnic groups. So these other marginalized ethnic groups that may or may not see themselves under active attack are going to respond to these future data breaches and say, heck, no, you're never touching my data. And so until we address this head on, will we really be able to move forward with this technology and improve the economy? Because if we look at short-term gains and neglect these future issues, then the overall cost, I believe, in the future will be higher than the amount gained by.
Do you want to share some of the polling results with us quickly before we move on to a couple of, I think, the final questions?
Yeah, so our question number 2, which was asking about the threat of genetic information, whether that is a near-term, midterm, or far-off threat. A majority of people thought this was a midterm, but only by a slim margin. A lot of people also equally thought that it was a near-term issue that needed to be immediately addressed. And then in our final poll, number 3, we asked, what is the biggest hurdle to implementation of genomic security policy? And a majority of people thought that the lack of understanding and awareness was the major hurdle towards making new policy. Also, lack of political will and need for technical expertise. So I think this really ties back into the conversation about having scientists in the room and raising the alarms on these potential issues.
We have some very smart attendees, I think, and I just want to speak to the first one where midterm to near term, it's exactly how I see it. Split in between the two, how quickly could this come out and when will it start affecting, who will it start affecting first? But I have to say that from my unofficial polling in the past, that has changed from pre-COVID when it was almost completely dismissed as being speculative of long-term threats. So just showing that maybe when we're saying, okay, let's be positive and try to hope it's just a midterm threat, we could be surprised, say, in the near future to have that become not a far-term threat, but something that we can deal with.
So we have a couple of questions. One of them is how you speak to people who are looking at contributing their genetic data to some of these databases. So one of the questions is, what advice would you give to consumers who are looking at genetic testing as an activity which is often undertaken more or less for recreation right now, rather than for medical diagnostics?
So John Sotos was asked the same question at DEF CON 25, which was 2017, by the way. Everybody has a personal choice to make, and everybody has something to be gained in that choice. They may want to desperately find their parents, for example, or find a long lost relative. And that might be worth all the risks in the world. And so I would never tell everybody that there's no way that you should ever do some type of test that can find a relative. I would recommend that you do your research on the company that does this. I would also recommend strongly against putting your information out there publicly. I do know that some companies, like I've seen actually from my experience with Ancestry.com, that you can use their services and then delete your data afterwards and actually have your data deleted. Other companies, maybe not so much, right? Other companies might have a business model of selling that data. It might have all these complicated terms of services. Now, I can't say that that's the current case with Ancestry. I just know that, hey, you can find relatives with it. And I have seen people before delete their data, right? Is there an easy solution here? No. And the answer that I give is that I certainly would not participate in anything until we have strong cybersecurity protections in this area. I'm not even generating my data. I would love to have it. But again, people might need this for medical reasons. They might need, their doctor might say, hey, you live or die, you give me your genetic data. At that point, maybe you should consider whether or not you want to participate in one of these research studies. Again, I'm not impressed by what I've seen with the way that the data is handled. And I would point out that a lot of these research studies, again, everyone has the best intentions. So they start out and you want to participate because you want to help science, and the researchers want to solve these diseases, and they all get together and they collaborate and they tell you, okay, we have the strongest cybersecurity, but one breach, the data's out there and it's out there forever. So your age plays an effect here too. Younger people have a different horizon than somebody who's older. Yeah, I wouldn't do it. So if that answers that question.
No, I gave the same advice to my father, who promptly ignored it. So they're so tempting at Christmas.
I know. $50, it's so cheap.
Exactly. We have an interesting question about the increasingly used term cyber biosecurity versus cybersecurity. One of our attendees has asked you to consider, is there a difference between cyber biosecurity and cybersecurity?
Okay, terminology, it always gets so complicated. And then cyber biosecurity, what does that mean?
Yeah.
You know, is it biosecurity for cyber or cybersecurity for bio? Let's assume it's cybersecurity for biology. There are certainly nuances that one needs to consider when doing cybersecurity for biological laboratories. You could claim that cybersecurity is cybersecurity, and I would agree somewhat to that. But if you're operating an industrial system where machines are constantly talking to each other, you have a different environment than if you're operating a business where you have, you know, machines connected to the internet that are constantly talking or else something explodes. So every cybersecurity environment is unique. We need to consider that biological environments are unique. I would even say we could break that down and say, well, there's biomanufacturing, there's hospital systems, there's genetic sequencing laboratories, and that these are all very unique, and that there's a great emerging field of cyber biosecurity, and that's awesome. But in a way, it's just a term. Are we hurting ourselves by creating a term that causes us to pigeonhole researchers? And you know, maybe we just want to call it cybersecurity. But that's.
Thank you. I'd say the terminology is tough.
Yeah.
Particularly in a place where there's already not a great deal of widespread understanding of what the problem is, and then a further narrowing for different technology, different terminology for that.
Yeah.
Thank you. So we have a couple of open-ended questions, and want to give you time to think about those, and then if you a minute or two, so that you can give whatever closing remarks you would like to offer, and then we'll close out our session today. One of the questions is really looking at, actually a couple of the questions are really looking at the same approach to determining what the threats are. So I'm going to paraphrase multiple questions into what is it that keeps you awake at night? You're talking about a short-term to mid-term threat, and obviously when we're talking about biological weapons, we can think about how older technologies have been deployed in ways that would have had bad outcomes or were intended to create stockpiles. What is it that keeps you awake at night in terms of beyond the individual privacy issues the WMD faces?
I mean, the really scary stuff, right, gets into sci-fi land, and it's not certain. And so I don't dwell on things that are so uncertain. Because if I was to just think, oh, well, this could happen, this could happen, this could happen, and they're all not really things that are worth me staying up late, losing sleep over. Instead, I actually lose sleep over where we're at today with the current status quo on this. Because if we continue to get stuck with, well, the data's worth money and we need to advance science, we're setting ourselves, you know, as a country far behind other countries that actually have controls around this. And so how do we get over that issue of, well, scientists like open data and the data is valuable? You know, how do we get from there to actually doing things to protect the data? And yeah, that's the biggest challenge I see. We need to start small, just like with lab cybersecurity, and take steps towards the goal. And when nobody wants to move at all because they have this claim of sort of you know, overarching science needs to be done openly and these things, I agree with all of these things, but you don't want to harm participants. And when that harm is theoretical, then it's easy to dismiss it. And so, you know, I'm deeply worried that we're going to have to see no longer theoretical harm before we start taking action on this. And at that point, you know, how bad could that be? I don't want to speculate, but we need to move. It's like go do something at all. Like at least require scientists to actually do security to actually be trained in security before they can claim that they have cybersecurity, and don't just let them check the box. Like just little tiny things that are not being done, unfortunately, right now. Need to be, somebody needs to give a little kick.
Well, and that actually leads into a second and related question, which is about the threat actors in this space. So as you're talking about what the vulnerabilities that are there right now that we're not thinking about, who are the threat actors that are potentially capable of hacking, or who might be involved in hacking, and what are the different postures right now that are in place in terms of defenses against, say, an individual actor, a rogue actor, a state actor, a disgruntled person within a laboratory. So who are the threat actors that you're thinking about when you're putting together the recommendations and the frameworks and the tools to provide protection for genomic and genetic information?
Well, obviously with the molecular cryptography, that's, keeps it off of computers. So you can imagine that I'm not just thinking about some random kid with a computer who would have trouble finding out where the laboratory is on a map, perhaps. But so yes, the technology that we're building is geared towards the most challenging of attackers. In general, it's with security. It's like, how much money do you have to put into security? What are the threats? What's the cost of something? And we went over those institutional costs. As I mentioned, the institutional costs are not the totality of the cost. The cost is borne by the people who gave the data and by the nation in which the data comes from which the data comes. So, when you're thinking of guidelines, you have to, as an institution, make those decisions based on all of those different factors. And who do I think about? I just think, let's build the best safe that you could possibly build, and teach people how to use a key properly, and do the best that you can. I hope that answers the question. I don't know. I mean, I don't know specifically like who are the hackers. Like good luck figuring that one out at all, right?
So I think one more question, then we want to make sure we give you a few minutes to share your closing thoughts. One of the questions was about how to make a better alliances between the organizations that are building toward, again, responsible use of genetic and genomic data for improving health, which is an enormous issue about equity, who benefits from data, who is contributing, who benefits from data, and how to build a conversation between those kind of groups. What the example that was specifically given was the Global Alliance for Genomics and Health, which has been one of the more active groups in talking about responsible data use for genomic data writ large and genetic data writ large. How do we promote a more lively and active dialogue between those groups that are focused on equitable use of genomic data to improve health globally, and the scientists who might have perceptions that they can share about the risks of sharing data that is individually identifiable? How do we promote a better dialogue between those two groups, both of whom ultimately have similar goals, right? Responsible use of genomic data to improve health, but are approaching it from very different angles. How would you promote a more lively dialogue about interface?
Such a tough question. So left the tough one for the end.
Sorry.
There. No, it's good. It's good. Because I just be like, I don't know. No.
And thank you all for joining us.
Yeah. We've run out of time. Yeah. So getting the participants involved is absolutely critical. For example, there was a study done on some indigenous Native American populations in the United States. They were researching one thing, the tribe was unhappy about what they ended up publishing. The problem I saw there was, well, there wasn't a communication between the scientists and the people they were studying, right? Because if they had said, hey, we're going to study this and we're going to publish on this, then they would have known ahead of time, no, that's going to make them very unhappy. So having that communication with people, making people informed. What is informed consent? You could think, right? Well, how do I inform a participant sufficiently so that they can consent when the threats are not necessarily entirely clear? But you need to at least have information going between all the different parties involved. And then because we're talking about some very serious threats, we need to have participation from ethnic groups that feel like they might be under threat, even though they're not even, no, well, they're only 2% of the study. Well, that's maybe enough for them to be under threat. And then we need to have security people and security-minded people involved to say, well, you know, these things can turn really nasty. At least this may be a threat in 20 years. And then get lawmakers involved. So what we really need to do is get all the people around the table and just start having this conversation and not just leave it between researchers who, well-intentioned as they are, could perhaps get into a little bubble and say, okay, we've worked all of this out, and they just don't have the expertise and they don't know, what's possible in regulation to enforce the people to do the right thing. So yeah, at this stage, conversations with many different parties is what's required.
Thank you. We actually still have a lot of participants very eagerly participating, but we are getting near to our end of our session. I do want to make sure before we close to give you just a few minutes to offer any last thoughts. Is there anything you'd like to leave with our participants today to think about as they see this issue evolving in the next months and years?
It's not ready to make closing thoughts, but
That's
that's
okay. Just
okay.
if there's any last thoughts. you
I
want to
do
share.
have a few last thoughts. First,
Okay.
first, I want to thank everyone for coming. This was a relatively large attendance. I think many people are looking at this from other countries as well. So I'm very happy that I could share this work with you. The big key takeaway is the data is not anonymous. So stop pretending like it is. That is a major annoyance that I have, and I've had for many years before the Golden State Killer got caught. I was telling people, hey, that data is not really anonymous. We got to be careful how we share it, and everyone's dismissive. At this point, there's no excuse. So stop telling people it's been anonymized. The data's been taken off of it, and then go from there, right? Then you can start talking about bioweapon threats. But the privacy issue, the informed consent that's required for researchers to share with their participants, please start there and do what we can to get regulation in there, to get the institutions to act properly. When we start talking about these potential bioweapon threats, those are very serious threats. So once you get over okay, it's not anonymous data, then we need to start moving into, okay, what are the real threats that we're going to be facing? When are we going to be facing those threats? And what can we do to protect the data and mitigate those threats? And take the problem seriously and get everyone around the table, get all the different parties involved, at least hear their opinion. Let's get this moving, because if we continue to neglect this, then 10 years from now, it could be very well too late if almost everybody's DNA is out there and it's essentially on the black market. Then there's not much that we could do to mitigate all of those threats that I discussed. And with that, thanks again for participating, and thank you very much for CRDF Global for hosting me here. You guys are awesome.
And I'd like to, on behalf of CRDF Global, to thank you for your time and for helping us have this really thought-provoking conversation. Like to acknowledge that in our online participation, we have a lot of country partners who have been long-time partners with CRDF Global about thinking about how to anticipate biological risks and implement biosecurity that's appropriate for laboratories at every level, and that can be implemented at the national level as well. One of the things we pride ourselves on at CRDF Global is working from the global to the national level, and then from the national level down to the institutional and laboratory level, where everything comes down to the commitment of individuals to make the work that we all do as safe and secure as possible, so that we can enjoy the privilege of conducting research and improving our knowledge and tools to keep advancing human health, to keep better understanding and protecting our environment, and to keep moving our technologies forward for a better future. So I'd like to take a moment to just appreciate the time you spent with us today, to thank all of our partners who have spent 2 hours online and continue to hang in there and ask questions.
Good questions.
Yeah, time in cyberspace is somehow always 5 times longer than any other kind of time. So it's certainly a tribute to how interesting the conversation has been that they've stayed with us. So thank you again for their time. Thank you to CRDF Global for your time putting this on, and to the security assistance team who has put a lot of thought into making this event possible and such an interesting conversation.
And maybe one last thing I would say, if you want to do something about this, contact CRDF Global, because there are things that we can all work together on to start addressing this problem. So I'll do that plug for CRDF, that maybe you were too modest to do it with that, so we can end there. Thanks, everybody.
So thank you very much to everyone.